This is Dr. Veena Jasuja here, and today we are going to do the test for color vision. Uh, it's examination of a second optic nerve. We had done the visual acuity in the last practical, and today we are going to do the color vision. So we have two types of photoreceptors in our eye, cones and rods, and the cones are responsible for the color vision. There are three types of cones in our eye, and these three types have three different types of photosensitive chemicals in them. And the sensitivity of these chemicals are different. Each chemical is sensitive to a small range of light. There is a trichromatic theory, and according to this theory, there are three basic colors: blue, red, and green. And you can perceive the different shades color spectrum by different combination of this basic color. So when the eye is stimulated with particular color, the three different types of cones are stimulated but to a different extent. So ratio of this red, green and blue is responsible a particular color your CNS is able to recognize. So let's understand this by this example. Suppose a orange light which has a wavelength of 580 nanometer, it is stimulating the eye. So here your red cones are stimulated to the stimulus value of 99. So 99% of peak stimulation at optimum wavelength. Then your green cones are stimulated to the value of 42 and your blue cones are not stimulated at all. So the value is 0 and the ratio of stimulation of three cones are 99, 42 and 0. Red 99, green 42 and blue 0. So the nervous system will interpret this set as an orange color. So likewise, so this is the way different ratios of the stimulation, and you interpret that particular ratio as a particular color by the nervous system. So that is the mechanism of color. Now, what are the different deficiency in color vision? Either a person is unable to distinguish particular color, or one or more of the cone system is missing, or there's some defect in a particular cone system. There may be a abnormal color matching, and there may be a confusion about the color. So maybe a marked reduction in the number of separate colors that can be distinguished in a particular that spectrum. So these are the different color vision. And to test this color vision, as far as this particular occupation are concerned, is very important. Suppose, for example, a person working in a textile mill, or handling the garments, painting mills. So this particular industry, if their color vision is not proper, they are not able to do their job properly. Same way armed forces, commercial drivers, and think about the railways, that the person who's giving a signals, if that person is colorblind to red and green, can you imagine what will happen? So is this like occupation, certain occupation, the color vision plays an important role, and their color vision is being checked before giving them a job. So what are the different types of color vision defects? Um, if any one cone may be a deficient or can be absent totally. For a normal sight, trichromatism, all the three cone systems are perfect. There's no deficiency, they are not absent. Anomalous trichromatism, they can differentiate all the colors, but the sensitivity is either reduced or displaced. So first, throat anomaly. Anomaly means weakness, okay? So throat anomaly, the color is red. The cone for a red color are either they are not able to do their job properly, they are deficient. And for Deutsch anomaly, the green cone is responsible. And last, red anomaly, the blue cone is deficient. So anomaly means weakness, rot anomaly, red color, Deutsch anomaly, um, green color. So red and green color anomalies are common. Blue color is very rare. Red anomaly is quite rare. And dichromatism, here there's a one cone system is absent. The same like anomaly, the names are the same, but anopia, there's an absence of one particular cone system. Trat anopia means blue cone system is absent. Tut anopia means green cone is absent, and prot anopia means red cone is absent. So monochromatism means the totally they are unable to differentiate colors of equal brightness. Now, there are particular few tests we perform in our physiology lab for color vision test, and uh, the aim is to identify particular deficit to classify and grade the severity of the color vision deficiency, and for a particular whether the person is suitable for a particular occupation that you can determine with the color vision test. So here we are going to perform, I'll demonstrate you this test later. One is pseudo isochromatic test plate. Sorry, we have a Ishihara plates in our lab. I'll show you that next. Then there are few tests of color naming and color sorting. We have Edridge green lantern in our lab, lantern test, and we'll do the home greens color test. So first of all, there should be a proper light, daylight, there should be a proper illumination. You'll explain the test to the patient, and patient should use his her near correction glasses if he has, and avoid the tinted spectacles or contact lenses. So suppose a person is illiterate, there's some pattern in this particular plate, and you tell that person to trace the pattern. So this is the way you check the color vision with the help of Ishihara color plates. Next is lantern test. Here there are different plates for different colors. You do it in a dark room, and you can create the different atmospheric conditions with those help of those plates. I'll demonstrate this in a video which will be shown you later. So this is the one the ophthalmologist. Then the Wormgreen's wool test. Um, Again, here you have a pile of color yarns. You tell a person to pick up a particular color from those collection of the wool, and either you can tell them to match the color. This is not very effective for diagnostic purpose. These yarns fade and they become dirty with the handling. This, before I demonstrate you how to do this test, let's talk about the applied aspects. Most common defect is red-green color blindness, and this is a genetic disorder. There's a lack of particular color gene, and this gene is present on X chromosome. And so that's why the lack of color gene is a recessive trait. The females are the carriers, and the males mostly suffer from the red and green color blindness. And so this is about the red and green color blindness, and now it's time for the demonstration. Okay, let's go for that. Hello everyone. Today I'm going to demonstrate to you all the test for color vision. There are three tests we do in a physiology lab. One is Ishihara's chart for color vision. Then we have a wool test, Holmgren's wool test, and third we have a Edrich green lantern. I'll show you that instrument later. First, I'll show you the Ishiara's plate. There are different color plates like this. In background, you have a dots of particular color, and there is a different color. There is a one some figure is written. You can see there are few plates like this. Okay. So first I'll demonstrate you how you check color vision in a particular subject. Sureshbhai, please અહીંયા આવો. છે એમાં ડોટ્સ છે અમુક કલરના અને અમુક બીજા એક કલરથી કંઈક લખેલું છે એ તમારે વાંચવાનું છે. આમાં શું લખેલું છે? 12. ઓકે. હવે આમાં
57.
ઓકે. Okay. Okay.
74.
So he is able to read whatever figure is written. In a standardized chart, that figure is written here at the bottom. Now suppose this person is illiterate, then we have a different place for that. X is written here and X is written here. In between there is some wave, you can say some pattern is there, and we'll tell the subject to follow that pattern with the finger. આમાં ફિગર નથી પણ કંઈક પેટર્ન છે એ આંગળીથી તમે આમ ટ્રેસ કરી શકો છો. આમાં ટ્રાય કરો આંગળી મૂકીને. So these are the Ishiara color plates in which in the background you have a dots of particular color, in which on top of it there's a different colors figure is written, and that figure is written at the bottom. And that person should be able to tell you that figure within 3 seconds. For the tracing of the thing, you can give a person at least 10 seconds. If a person is color blind or color weakness of particular color, that person will not be able to read that particular figure, or he or she will read something else. So this is about the Ishiara's color plates. This is Holmgren's wool test. You have two sets of the wool patterns here, and you tell a person to pick up the same color from the second plate set. So Suresh bhai, આમાં જે કલર્સ છે હું જે કલર પર આંગળી મૂકું એ તમે મને આમાં બતાવજો. આ કલર
Green.
ના એટલે આમાં બતાવો મને, આંગળી મૂકીને બતાવો. ઓકે. આ રેડ કલર આમાં ક્યાં છે? ફાઇન. ધીસ. Okay. We have here the two sets like this, or you can have a box in which you could put these yarns and tell a person to pick up the particular color from that box. So this is a wool test. It's green lantern. This test they used to do for the drivers in the railroad. That is it's so ancient instrument. You're supposed to identify this and you should know the principle. Have a look here. There are different plates here. The first one, you can change the diameter of this aperture here. This one, have a look. You can see the color green right now. There are different plates with different colors. So you can change the different color, and this has to be done in a dark room where you display this. Person is sitting at the 6 feet distance, and he or she should be able to identify the different colors. And this last plate is to simulate the atmosphere. Rain or the clouds or the sunny atmosphere, and that is the reason this test used to be done for the drivers where they have to drive in a different atmospheric condition. So just remember 3 tests for color vision. First was Ishiara chart plates, then was a wool test, and the last is this lantern. So that is about the color vision. Thank you.